Hi everyone, my name is Brittany Fischer, and I recently obtained my master's degree from the Department of Animal Sciences at The Ohio State University. Today I'm sharing with you my project, which was the development of a longitudinal welfare approach for cheetahs in human care. This project was made possible by the partnership of the Center for Human-Animal Interactions, Research and Education, better known as CHAIRE, and the Columbus Zoo and Aquarium. First, here is the definition of animal welfare that's followed by the Columbus Zoo. It's defined as an individual animal's collective physical, mental, and emotional states over a period of time, which is measured on a continuum from good to poor. Animal welfare was established as a science just over 50 years ago, and it began due to societal concern for animals in human care settings, and since then has developed into a multidisciplinary field combining experts from various backgrounds. Within the zoological industry specifically, advances include a new animal welfare accreditation standard from the Association of Zoos and Aquariums, or AZA, which the Columbus Zoo is a member of, animal welfare research centers, and animal welfare monitoring tools. Various frameworks to measure animal welfare have also been developed. The 5 domains model is most utilized by zoos and includes the domains of nutrition, environment, health, behavior, and mental state. Through literature review, several gaps were identified to shape my project specifically. First, studies often use one measurement for welfare. Second, studies often involve multiple zoological institutions. And lastly, measurements are often of acute or short-term stress. Behavioral research is one measurement that allows for objective measurement of an animal's outward expression of how they're coping within their environment. One limitation of this measurement alone is it does not provide information about an animal's stress level. Cortisol is a metabolic hormone that's involved in the stress response. Hair sampling to measure cortisol production reflects long-term cortisol concentrations as an indicator of potential chronic stress, so different than those acute or short-term measurements. For my project, the overarching goal was to enhance the 5 domains animal welfare model by combining hair sampling and behavioral observations to measure welfare of cheetahs in response to changing environmental factors. There were 11 cheetahs in my study population of various ages, sexes, and housing occupancies. Some cheetahs participated in a cheetah run activity to promote instinctual chase behavior, which I hypothesized would affect their behavior and cortisol levels. My study was separated into 6 60-day periods, and the exhibit was not open to the public year-round, which I hypothesized would also influence welfare. The cheetah run activity I mentioned previously did also not occur year-round. The first 3 domains of the 5 domains model are resource-based, and therefore I verified each domain by use of species and individual histories, including medical records and interviews with staff members. I used a scan sampling technique for my behavior observations, in which I recorded behaviors every 5 minutes for 2 hours at a time for a total of 150 observations per period. I also used an ethogram to catalog behaviors. I took 1 hair sample at the beginning of each period to align hair growth with behavior observations. Cheetahs were trained for the collection using positive reinforcement, and the same sample location was used each time, as you can see in the image there. I analyzed my data at the group and individual level. For the group, we found that cheetahs were more likely to perform stereotypic behavior if they participated in the cheetah run, were housed as pairs or by themselves, as well as during periods 1, 2, and 3. This graph demonstrates averages of stereotypic behavior using least square means of all 11 cheetahs across all 6 periods. As you can see by the superscripts, many cheetahs differed from each other throughout the study. We did not find differences of cortisol concentrations at the group level, but further analysis at the individual level are displayed in this graph. As you can see, cheetahs 1, 7, and 10 showed increased concentrations compared to other individuals. This table again shows those 3 individuals, as well as an effect of the absence of the cheetah run activity. Although the population as a whole was able to adapt to the absence, cheetah 10 showed increased concentrations during those periods. Overall, my study demonstrated the importance of a multifaceted approach to assess all domains in the model. Use of hair sampling was appropriate, as well as assessment at the population and individual level to demonstrate how individuals differed within their changing environments. This study can be implemented within other zoological institutions to promote evidence-based management decisions. My approach can be used for other cheetahs and other species as well. And lastly, it's important to translate this information to the public. Thank you all for taking the time to listen to my presentation and to CHAIRE for allowing myself and others the opportunity to present our research virtually.